Human papillomaviruses, HPV, are double-stranded non-enveloped viruses that have a diameter of roughly 55 nanometer. This virus can enter the epithelium through disruption of the skin or mucosa and infect basal stem cells. This virus has three functional coding regions: early region, late region, and long control region, also known as the non-coding region, which lies in between the early region and the late region. Those who have several sexual partners and/or persistent HPV infections are particularly vulnerable to acquiring additional HPV subtypes. HPV is known to be the main cause of cervical carcinoma. There are two types of HPV, low risk and high risk. For those who have a low risk HPV infection, it can lead to the development of warts in areas such as the genitals, anus, mouth, or throat. The HPV types 6 and 11 are in the low risk HPV class. High-risk HPV infection itself is asymptomatic. However, if the infection persists for years, it can lead to precancers and cancers at the infection site, potentially causing symptoms like lumps, bleeding, and pain. The type of HPV that is considered a high-risk HPV type is HPV type 16 or 18. For prevalence and incidence, statistical and geographical analysis indicates that it is more prevalent among Native American women than Caucasian women. According to statistics from the World Health Organization, WHO, around 500,000 new cases are reported annually, with 250,000 of these cases resulting in fatalities. This concerning trend has prompted molecular virologists worldwide to delve deeply into the pathogenesis of papillomaviruses and explore potential therapeutic solutions. The next section discusses the pathogenicity of the human papillomavirus. First, we need to understand the fundamental structure of HPV-16. There are various types of viral protein on HPV-16. Each of that has unique purpose. So E1, E2 role is to code proteins that control the function of the E6 and E7 genes. E4 control viral release from the cell. E5 is used to enhance cell immortalization. The tasks of E6 and E7 are to inhibit and degrade tumor suppressor genes, P53 and PRB respectively. So now the question is, how does cervical cancer develop? What we get here is actually the close-up image of the human cervix. HPV infection can get inside our body through a cut or wound at the cervix. Then this virus will invade the basal cell layer and then start to give infection. Within persistent infection, E6 and E7 will play their role in this moment. E6 viral protein will bind and degrade the tumor suppressor gene, which is P53, that functions in the apoptosis process. So the infected cell cannot undergo normal programmed cell death or apoptosis. On the other hand, E7 will also bind and degrade another tumor suppressor gene, PRB, which plays a role in regulating the cell cycle. In the absence of PRB, the infected cell becomes proliferated and uncontrollable. To sum up, E6 and E7 are responsible in order to make the infected cell aggressively proliferate and progress to become a cancer cell. Risk factor and causes. Risk factor is not 100% that you can get the HPV infection, but it has high potential risk. Firstly, you can get HPV through sexual activity. The virus can transmit through intimate skin-to-skin contact when unprotected sex happened. Age can be a factor because late teens and early 20s are in high-risk group due to new sex partner. They also have less information about the risk and has the urge to try something new. Next, oral contraceptives can alter the hormone levels and affect the immune response. The ability of the immune system to fight HPV is weakened as a result of hormone imbalance. Smoking also has a risk of developing cervical cancer. The tobacco substance inside the cigarettes has the potential to harm the cervix. Lastly, compromised immune systems may be the cause of our body's weaker ability to defend against an HPV infection. For example, patients who are undergoing chemotherapy struggle to eliminate the virus and may develop complications such as genital warts and cancer. Differences between benign and HPV-infected epithelial squamous cell. Knowing the characteristics of a benign intermediate squamous cell is as important as knowing the characteristics of an infected cell, as it helps in distinguishing between normal and abnormal cell. The most crucial aspect when observing a cell is to look at the characteristics of the nucleus itself. A benign intermediate cell will not have any atypical nuclear enlargement, which is more than 3 times the normal nuclear size. The cell should also have a low nucleus to cytoplasm ratio as the nucleus is much smaller in size compared to the cytoplasm. The nucleus of a benign intermediate cell should also be centrally located within the cell, as well as having a smooth and thin nuclear membrane. The chromatin pattern is also one of the most important indicator in knowing the status of a cell, where a benign intermediate cell will have a chromatin pattern that is fine and evenly distributed. Other than the characteristics of the nucleus, the cytoplasm can also be observed to determine whether the cell has normal or abnormal characteristics. Polygonal shape is typical for a normal intermediate cell, with the cytoplasm usually appears as thin and translucent and is usually cyanophilic in color. There is a clear difference between benign and HPV-infected epithelial squamous cell. On the characteristic of the nucleus, we can see that the nucleus are enlarged more than 3 times, irregularity of the membrane of the nucleus, high nucleus to cytoplasmic ratio, multinucleation, which is a cell with more than one nucleus, coarse and not evenly distributed chromatin, and hyperchromatic color of the nucleus. For the characteristic of cytoplasm, we can observe the formation of koilocyte, which is a large cytoplasmic clearing and a very dense cytoplasm. Relationship between HPV infection and koilocyte formation. What is koilocyte? A koilocyte is a term used to describe cells that exhibit changes in characteristic of being hollow. The term koilocyte is derived from the Greek word koilos, meaning hollow. These changes are important in the field of pathology as they help in identifying certain medical conditions. Koilocytes are a distinctive indicator for diagnosing low-grade squamous intraepithelial lesion, or LSL for short. It is a type of abnormal growth of squamous cells on the surface of the cervix. The presence of koilocytes can help pathologists determine the presence of LSL, which is a crucial step in the early detection and management of cervical cancer. Now, let's discuss the morphological characteristics of koilocytes. One of the key features of a koilocyte is a ring of dense cytoplasm that typically encircles the perinuclear cavity. This creates a halo or clear area around the abnormal nucleus. This halo effect is one of the distinguishing features of koilocytes and helps in their identification under the microscope. Lastly, it's important to note that koilocytes are an indicator or marker of HPV infection. Human papillomavirus, or HPV, is a common sexually transmitted infection that can lead to various types of cancers, including cervical cancer. The mechanism of koilocytosis is similar to the pathogenesis previously explained. HPV produces several proteins known as oncoproteins. These oncoproteins are responsible for the oncogenic process leading to cervical cancer, as shown in the picture. E4, E5, E6, and E7 are the proteins responsible for this mechanism. First, HPV infects the basal cell through a cut or lesion. When HPV resides in the basal cell, it transcribes its DNA to form viral proteins. E6 and E7 cause nuclear enlargement, irregular nuclear contours, and hyperchromasia. The E4 protein disrupts the cytoskeleton of the cytoplasm by binding to keratins, thus forming the perinuclear cavitation. E5 and E6 also work together to form the perinuclear cavitation. As for the diagnosis of HPV infection, genital warts may appear on the vagina, penis, or anus, and rarely the throat. The warts may be painful, itchy, or bleeding. Survival cell changes also can be seen when doing cytological tests. The changes will lead to precancers that may become cervical cancer if left untreated. It usually takes 15-20 years for cervical cancer to develop after HPV infection. There is currently no treatment for HPV infection. Treatments exist for genital warts, cervical precancers, and cervical cancer. Prescription medicines include Podophylox for external warts, to stop the wart cells from growing, Imiquimod, boosts the body's immune system so it can fight HPV. For invasive cancer cases, the treatment includes surgery, radiotherapy, and systemic therapy, such as chemotherapy. HPV infection can be prevented through vaccination towards risk groups of the infection. Prophylactic HPV vaccines, such as Gardasil, is found to be effective in treating HPV infection. The vaccine should be given to all girls aged 9 to 14 years before they become sexually active. Next, avoid smoking as it makes the immune system less effective in fighting HPV infection. Lastly, use protection while doing sexual activities to reduce the risk of getting HPV infection.